Um, I want to appreciate you, Ayana, for saying that, though. That take a certain amount of vulnerability and bravery, but I would agree with you. I think the weather has something to do with that. I think everything that we've been processing from our news and from our social media has something to do with that, and the fact that it's a Monday. We're fresh off of a weekend, and so there's just some natural fatigue, I think, that comes with that. So I'm with you, though. I don't feel 100%, and we'll get through this lesson and kind of think through what else we can do. But I'm going to go run and get I'll start the presentation so you all can start your warm up. Then I need to go run and get my laptop charger.
I have a question.
Yes,
Is it
ma'am.
okay, like my hair? I just put it in a ponytail and I have like a scarf wrapped around it. Am I allowed to wear that?
Yes, that's fine.
Okay.
I'm sorry that you even felt the need to ask that.
Yeah, one of the teachers was saying no hats, no hoods, no hoodies, no other stuff. So yeah.
All I can do is shake my head to that. But no, you're welcome to. You're at home. We're all at home. You're welcome to wear whatever you'd like to wear in your house. It's your space. Okay. So the timer has started. You do have a warm up as you do every day. Unit 1, day 6. Go ahead and knock that out for me. And like I said, I will be right back. Okay, we've got 4.5 minutes left. Okay, so hopefully you all had an opportunity to go ahead and knock that warm-up out. Just as a brief aside, all of our assignments for this first unit will be due tomorrow, which is the last day of the first unit. We will have a quiz tomorrow. It'll be similar to the quiz we took, I think Thursday of either Wednesday or Thursday of last week. Like I said, the other biology teachers and I are thinking through some potential changes, to the quizzes so that they can be a bit more cumulative, because as they are currently designed, they only track one standard. Only one standard is tested per assessment. So we want to blend them a little bit so that you have multiple standards that are tested, as well as blend them so that there's some amount of continuity shown as the school year goes on. You can see how you are improving on each standard. Okay, so that quiz that we took last week, I will likely curve pretty substantially, so everybody will get a boost. And then the quiz that we will take tomorrow will focus primarily on enzymes, which we talked about on Friday, but we're also going to continue that discussion today. So we won't have much asynchronous work to do today. There's only one short assignment. So those 35 minutes, I would strongly recommend to you that you use that time to kind of catch up on anything that you haven't been able to do thus far. Many of our assignments, as you should have recognized by now, are very short, and so they don't take much time, but you do want to make sure you're staying organized and doing a little bit each day so that you don't get until the end of the unit and still have a lot to do. So first things first, I do want to, there's Angel right there. Hi, Angel. I do want to make sure that you all have an opportunity to share anything that's on your mind. I know Ayana has said that she's the only, she doesn't feel great today, and she is not alone in that, I'm sure, but does anybody want to share anything else that's going on? Okay, I'm going to take that as a no. And I saw Kiki, you got a nice yawn in, so we're all in that same place. We're all just a little bit tired today. And we're all starting to get used to, Stephanie, I was just wondering if anybody wanted to share anything about their weekend or about anything that's on their mind or anything that they're feeling. But I do want to emphasize that the classroom culture aspect of this learning environment is going to be important, but I'm still learning as the teacher how I can do that, because we're so used to actually being face to face and being able to see you walk into the classroom and you all can sit next to specific people and you start to build connections in that way. But virtually it's different. So we're still trying to figure that out, but that doesn't mean that it's any less important. So even though it's awkward to be talking to a computer screen and you can't even see most of the people's faces who you're talking to, that is something that we're all just going to have to push through. And hopefully we get to go to school eventually when it is safe to do so. All right. So we don't have an essential question for the day. I will briefly read your objective, which states that biologists will be able to understand how to read an enzyme graph and determine an enzyme's optimal temperatures and pH. All right, so again, we're thinking more about enzymes today, and we ended our last discussion with a brief introduction to this concept of optimal temperature and pH. We'll talk more about that today and also give you all the skills you need to be able to read a graph to determine where that optimal temperature or pH is. So you've done graphing in math, but graphing is also important in science as well. In fact, it's extremely important because as scientific research is done and as the results are collected, you need to be able to represent those results in a way that is understandable not only to you, but also to other people who might want to learn from your research. So these graphing skills are extremely important, and there are hundreds of different types of graphs and ways that you can represent data. But for the purpose of this biology course, we really only need to be able to read this basic line graph. So can someone tell me which color on this graph? I know some of the colors might be a little difficult to see, but what color on the graph represents the x-axis?
Yellow.
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 Excellent. Yellow. And the y-axis would therefore be
Purple.
purple. Good. So we can also call the x-axis the horizontal axis, and the y-axis is the vertical axis. All right, so you all know that. So we talked about this on Friday briefly at the end of the lesson. But enzyme activity can be graphed, and it will be graphed such that the enzyme activity is on your y-axis. So as I go up on the y-axis, what I'm saying is that the enzyme is working better. So as I look at this graph, as the red line gets higher, that's when it's telling me that the enzyme is working really well. So of course, the highest point on that graph is going to be my optimal environment. That will tell me where the enzyme is working the absolute best, wherever the graph is the highest. And then when the graph is at 0, of course, we're still on the y-axis here. When the graph is at 0, that means that the enzyme is no longer working at all. And we call that denatured. When the enzyme is no longer working, that's because it has changed shapes, and that's what we call denatured. So can someone tell me what the optimum pH level is here? Should be very obvious.
8.
8, thank you. Castro, I saw you hold up your fingers too. I appreciate you. Sorry, okay. And can someone tell me when the enzyme denatures?
Below 5 and above 10.
Good, below 5. So below 5. And then also above about 10 and a half. All right, so typically there will be 2. Gabby, I'm going to put you on mute. Sounds like there's something barking in your background. But typically there will be 2 points on the graph where the enzyme denatures, because it happens both in the more acidic range and then also in the more basic range. So this specific enzyme, as several of you told me, has an optimal pH of 8. That's where the graph is at the highest. And so that means that it prefers something that's basically neutral. But it can still work when I'm at a pH of 6. It just doesn't work as well. And it can still work at a pH of about 9, 9.5. It just doesn't work as well. But when I start to get lower than, let's call that 4.75, then that's a problem. The enzyme no longer works. And then when I get higher than 10.5, that's also a problem. The enzyme won't work anymore. All right, so you got to be able to pick out really just those 2 things, the optimal level and then where it denatures. And there should be 2 points of denaturation. Okay. So what's the optimum temperature for the enzyme shown here?
40.
40 degrees Celsius, yes. Now, in the past, we've really just looked at the pH, but the same graphs can be done for temperature as well. So the optimum temperature in this case is 40 degrees Celsius. At what temperature does this enzyme denature?
Like around 62 to 63.
Good, yeah, around about 62 or 63. Now we can't see where it hits 0 on this side. As Castro just told us, we could see at about 62 or 63, that's where it denatures. It doesn't work. But on the colder side, we don't see it actually touch 0. So in this case, we can't say that there is a second point of denaturation. We just have to rely on this higher temperature that we can actually see. Okay, brief review of what we discussed on Friday with enzymes as well. It says the diagram shows an enzyme substrate complex. Which of the following is represented by structure X? So here's structure X. Is structure X a substrate, or is it a product, or is it the enzyme, or is it a complex? What do you all think?
I think it's A.
It is A. Excellent. Thank you. I think that was, I'm not sure, was that Gabby?
No.
That was Alicia?
Yeah.
Okay, good. So yeah, structure X would be A. It's the substrate. So let's break that down a little bit more. The substrate is your reactant. It's what you are putting into the reaction. It's what you're starting with. All right, so we can see that these arrows are giving us some directionality. We're starting over here, and we're moving to this in step 2, and then we're moving to this in step 3. So in step 1, what we're starting with, we've got 2 things. We've got structure X and structure Y. Alicia told us that structure X is the substrate. Does anyone know what structure Y is?
Mr. Red,
Yes,
I know we're supposed to like keep our cameras on, but can I turn mine off?
that's okay.
Thanks.
Isn't structure Y the the complex? Or am I tripping?
It's not the complex, but okay, so the complex is, well, we'll talk more about it. But structure Y, if you notice something, it appears the exact same in step 1 as it does in step 3. So what should that tell you? Nothing happens to it in the reaction.
It's an enzyme.
That means it's an enzyme, yeah. Remember, enzymes are reusable because they do not change in the course of a reaction. When I put an enzyme in, I should get it out in the exact same way when the reaction is over. And that means that I can then put that same enzyme into the next reaction and take it back out again. Then I can use it again in another reaction and take it back out again, and so on and so forth. All right. So typically the enzyme, when you're, and I can almost guarantee you all will see a diagram like this, not only on tomorrow's quiz, but also on the EOC. So this is a good question to ask. Typically, when you see a diagram like this, the thing that is not changing from one step to the last step is the enzyme. Okay, so structure X is your substrate, structure Y is your enzyme. What is structure Z? We only have 2 other options.
The product?
Structure Z is your product. Good. So we've got to get our minds thinking that the substrate is what you start with, the product is what you end up with. So the product is what you produce. It's what comes out of the last step of the reaction. So structure Z is our product. So what we can see here is that in step 1, our substrate approaches the enzyme. In step 2, the substrate actually enters the enzyme. And this is what we call Adrian's point. This is what we call complex. The complex is when the substrate and the enzyme are connected. And that's where the chemical reaction takes place. Then as we go into step 3, the chemical reaction has taken place and we've produced something different. We started off with a substrate, we produce something that is slightly different. And in this case, the substrate has been digested. So I start off with 1 reactant and I end up with 2 products. It gets split, it gets broken down. So this is a good diagram to study for sure. And if you search enzyme substrate complex, I'll type that into the chat. If you just search that in Google, I'm sure you will come up with a number of diagrams that look exactly like that. Let me test it out. Yep. So as you can see, same thing here. I've got my substrate, I've got my enzyme, the substrate enters the enzyme, the chemical reaction takes place, and now I end up with my products. Same thing in this diagram. Same thing in this diagram. All right, so that is an extremely important and extremely common diagram, and I can almost guarantee you'll see it not only tomorrow, but also on your EOC exam. Okay, so more review. What do you remember about nucleic acids? What's the monomer of a nucleic acid?
A nucleic
Amino acid.
What's that?
Amino acid.
Amino acids are for proteins. Amino acids are the monomers of a protein. So what about nucleic acids?
Nucleotides.
Exactly, nucleotides. So again, we really can be focused on these, on the letters NuCl. We really should start making the connections between those letters. So nucleic acids are made up of nucleotides, and nucleic acids are located in the nucleus, which we'll talk about in our next unit. What do nucleic acids do for us?
Control like the protein.
Good, they control the production of proteins. Anything else that we want to talk about?
There are DNA and RNA.
The examples are DNA and RNA, exactly. Okay, so here's a monomer. This is a nucleotide. We've got a phosphate base, a 5-carbon sugar, aka a pentose sugar, and then a nitrogenous base. Does anybody remember what our 5 nitrogenous bases are? You can just give me the letters. I'll give you one of them. One of them is A. Does anybody remember any of the other 4?
Can you repeat that?
Yes. So nucleotides are made up of a phosphate group, a 5-carbon sugar, and also a nitrogenous base.
ATGCU.
Yes, exactly. So we have 5 nitrogenous bases. One of them is A, another is T, another is C, another is G, and we also have U. Okay, so those are our 5 nitrogenous bases that could potentially be a part of a nucleotide. Okay. Thank you for asking questions too. I appreciate that. So DNA, which is of course it is a nucleic acid, it can be found in different forms. And the 2 forms that we'll focus on here are chromatin and chromosomes. This is going to come up much more in depth in Unit 5 when we talk about heredity. But it's important to note here too, chromatin is just a decondensed form of DNA. So by that we mean it looks like a big plate of spaghetti, or it looks like a scribble on a piece of paper. There's no order to it. It's just randomly wound. Some areas are tightly wound. Some areas are very loosely wound. It's kind of chaotic. Chromosomes, on the other hand, which you probably have heard of, are condensed and ordered forms of DNA. So they're very tightly wound in these, in these X-like structures. The X-like structures are also bound in the middle. Does anyone happen to know how many chromosomes humans have?
Isn't it like 23?
Okay, so yeah, we get 23 from each of our parents, which means in total we have how many?
46.
46, good. Okay, but again, we'll talk about that in much greater depth in the future, but it's important to know because we need to be able to think, and I mentioned this last week, but we need to be able to think about the connection between DNA and proteins. And as Castro told us already just a few minutes ago, DNA, its primary function is to control the production of proteins. DNA is the instruction that, if you think about like putting together a shelf or a bed frame, DNA would be the instruction manual. The protein is your final product. Where DNA would be your recipe book, proteins would be your meal. Okay. Now, on each chromosome, and of course, humans have 46, different animals have different numbers, different organisms have different numbers of chromosomes, but humans have 46, a healthy human, I should say. There are some syndromes that in which a person would have more or less than 46, but a healthy human has 46 chromosomes, and on each of those chromosomes, there are many, many different genes. Okay, so a gene, and you should be writing this down. I'm sorry that it's not highlighted. I'll actually highlight it. Okay, so a gene is just a small section of DNA that codes for proteins. I'll give you time to write that down, and then I'll break it down for you a little bit. Okay. So we have DNA, and our DNA is made up of billions of those A's and T's and C's and G's. Remember, there are no U's in DNA. The U's are only in RNA. So our DNA is made up of billions of A's, T's, C's, and G's. But only portions of that billion are actually used. And even smaller portions are used for specific things. For example, the gene that makes my hair color would only be a very small portion of that DNA. I don't need the entire billion letter pack of DNA to code for my hair color. Just a small portion of it that might be only a few dozen base pairs long. So a few dozen A's, T's, C's, and G's long. So we have many, many genes that code for all of what makes us who we are. They create all of these proteins that make us who we are. And so with all those billions and billions of base pairs, there is plenty of room for mistakes. And I remember getting a question in the first week about mutations, but this is where mutations start to come into play. If a mutation takes place in one of these segments in a gene that actually codes for something that's important, then it could be problematic. But most of the time, a lot of these mutations are not taking place in segments of our DNA that code for anything. So they don't have ultimately any impact on your life. Okay, we'll talk again. This comes up a lot more in Unit 5. So again, I really like to think in the form of analogies. So if I think about my DNA, which is, again, those billions of base pairs, about 3 billion base pairs. If I think of that as the entire recipe book, then each gene would only be one actual recipe in that book. So if I have a recipe book that is for, let's say, vegetarian recipes, and there's a chapter on vegetarian soul food, there's a chapter on vegetarian Mexican food, there's a chapter on vegetarian Chinese food. So all these different chapters, and then even within those chapters, there are specific recipes. So in the recipe for vegetarian soul food, there's a recipe for vegetarian barbecue cauliflower. And then there's another recipe for vegetarian macaroni and cheese, right? So there are all these different recipes. Within that book, there are probably some recipes that I'll never touch, that I'll never use. And our DNA is the exact same way. There are portions of it that just don't ever get used. And there are some portions of it that are used a lot. And that code for maybe several different things. And each individual recipe in my recipe book is what I would call a gene. I don't need the entire book to make one recipe. I just need that page to make that recipe, or even that small portion of the page. Okay. All right, so here's where we make the connection. Nucleotides are the monomers that make DNA. DNA is then converted into RNA, or we should really say it is transcribed into RNA. That RNA is then translated into amino acids. And then those amino acids are the monomers that combine to make proteins. I'm going to actually change this word here. I want to say this isn't working. No, it's wrong. Rough day, rough Monday. Directions that. Okay, I'm just going to get rid of this one for now. So this is called the central dogma of biology. This is the biggest theme, the biggest idea of biology. And so the discovery of this concept really wasn't made until the 60s. So, for the vast majority of scientific history, we really didn't understand how this took place. And we really didn't come into an understanding of what DNA was until an English woman discovered it in the late 50s. And so it was at that point that we started to better understand this big idea. And it's only been about 60 years that we've been able to use this information to further our research. But nonetheless, this is really the central idea of how life came to be and how genetic information is stored and passed on. I don't want to do that. All right, so let's watch this brief video.
DNA is the instruction manual for how to build life. From microbes to plants to human beings, it defines us all. The complete set of instructions encoded in an organism's DNA is called its genome and is passed from parent to offspring during reproduction. Information is stored in DNA using just 4 types of molecule which occur in pairs. There are billions of these pairs, organized in a double helix structure that is both strong and compact. These pairs also allow each strand to act as a backup for the other, a remarkably efficient way to safeguard this precious genetic information. DNA folds into paired packages called chromosomes that are stored in the nucleus of the cell. Different species have different numbers of chromosomes. Humans have 23 pairs. Chromosomes contain many genes. A gene is a section of DNA that holds the instructions for a protein. Proteins are essential for life and perform a huge variety of jobs, from controlling the function of a single cell to determining the shape of a whole organism. Within a species, each organism has very similar DNA. In human beings, the difference between one person and another is a fraction of 1%. But it's what makes us individuals, giving us different facial features, hair color, and height. The uniqueness of our DNA can be used like a fingerprint to identify us with an extraordinary degree of accuracy. By reading DNA, scientists have discovered that we share sequences not just with our own species, but with every other living thing on Earth. Chimpanzees, one of our closest living relatives, share about 96% of our DNA. But we also have genes in common with fish, plants, and bacteria. Powerful evidence that all life came from a single universal ancestor billions of years ago. We haven't just learned to read the instruction manual for life, we can rewrite it as well. People have been manipulating DNA since before we knew it existed, selectively breeding plants and animals to bring out desirable traits. Now, genetic engineering allows us to directly alter DNA in a lab, creating new varieties of life. From plants that can resist disease or drought, to bacteria that can mass-produce life-saving hormones. But we don't yet know what all of DNA does. Lengthy sequences make no proteins at all, and have perhaps mistakenly been labeled junk. Some people are worried about these gaps in our knowledge and unforeseen problems they believe genetically modified organisms may cause. What's clear is that the instruction manual for life is more subtle, elegant, and complex than we could have possibly imagined. DNA has revealed many of its secrets, but we still have much to learn.
Okay, any questions based on that? Oops, what did I do? See Kiki saying yes, no, maybe so.
No.
No.
Interesting. Okay, I thought for sure that would pique some interest and get you all to ask some questions, but that's cool.
It was trippy.
Well, do we know who our first ancestor was?
Do we know who our first ancestor was?
Or like what it was? Or when? Or anything?
We don't know. You know, we cannot prove that, but there is strong evidence that all life originated in the ocean, and with just a single-celled organism, and over many millions of years developed to what we know as life today, which is many millions of species existing in vast arrays of environments. But the original form of life was, first of all, it was anaerobic, meaning that it did not require oxygen, but it did produce oxygen as it was creating energy. And so once it produced enough oxygen in the atmosphere, that's when life began to become aerobic. So we'll talk more about that as well. That comes up in, I think, Unit 8 of this course. We'll talk about the origins of life. But like you may have heard him say that the fact that we share DNA, portions of our DNA with every living thing on Earth is strong evidence of the fact that we do have a common ancestor. And so we'll think about all the different ways. I can even show you one right now. All the different evidence that we have that we do have common ancestor. We do have a singular common ancestor. One of the other ways we can think about it is the fact that many different species essentially look the same as embryos. So if you look at this, there's virtually no way you can distinguish between a fish, a salamander, a turtle, a chicken, and a human as an embryo. We all look essentially a lot the same. And then as we continue to develop, inside of the womb, we become differentiated, but for those first few weeks, there's almost no difference. In fact, humans even have slits for where gills may have once appeared. So that's just some evidence of the fact that we all come from some earlier ancestor. Okay. Good question, Adrian. All right, so that's it. We have 30 minutes left in class. Like I said, the asynchronous assignment for today is very short. So if I were you, I would just go ahead and knock it out and then maybe work on something else that you haven't. You should probably be able to get 2 or 3 things done today in these 30 minutes. But I will be here. I'll mute myself. You should be working on the enzyme practice on Canvas. Then let me know if you need any help.
Mr. Rudd?
Yes, Gabby.
For number 13, it says what is the optimal pH for is it supposed to say papain?
Give me 1 second. Number 13. Okay, let's see. Yeah, so I'm not exactly sure how to pronounce it, but PAPAIN.
Yeah.
It's that last, like if it's the graph that's in the bottom right. You see that?
Oh, okay.
Okay, people, it's 11:35 now, so that means that class is over. If you have any questions or if you need anything from me in the next 23 hours, please don't hesitate to reach out. You will start to see some changes in your grade book, hopefully today, if I can stay on top of my work. So let's just all hope that remains true. But otherwise, you are good to go, and I will talk to you tomorrow.
I have a question.
Bye.
Yes, ma'am. See you. Have a good day.
I haven't turned in all my work yet, but I can still turn it in before tomorrow, right?
Yes, ma'am.
Have a good day.
You too. Thank you.
Bye.
Bye-bye.
All right, Mr. Rudd.
All right, gentlemen. Have a good day.